Hi everyone, my name is Kayla Pehl, and today I'm going to be talking about the relationship between locomotor performance and habitat use in six-lined race runners, Aspidocello sexlineata, in coastal South Carolina. How organisms are distributed in time and space is a fundamental question in ecology. A critical determinant of this distribution is an animal's locomotion because it affects defense, dispersal, finding mates, foraging, and escaping from predators. Habitat can affect an animal's locomotion in a variety of ways. The structural complexity of a habitat could impede locomotion due to obstacles in substrate type. Habitat could make movement more apparent and thus riskier. It could supply a greater density of resources, thus requiring a slower speed of animals, they can utilize them. And habitat can also provide cover from predators, which reduces the use of locomotion required for escapes. Since an animal's ecology is impacted by its habitat and habitat affects its locomotion, locomotion is often a critical component of an animal's fitness and survival. Lizards are common test subjects in locomotion studies because they express a wide range of locomotor behaviors and abilities. The goal of this study was to examine the relationship between locomotor performance and habitat use in six-lined race runners to see how they are impacted by variable substrates in a coastal setting. I had 3 research questions for this project. Do race runners move through their habitat randomly or non-randomly? How does available habitat affect their locomotion? And is the pattern observed in question 1 explained by question 2? I had 3 different sets of data for this project. First, I conducted a habitat assessment on the sand dunes on Sullivan's Island using transects. I collected data from 197 transects, each of which was 20 meters long, and I randomly crisscrossed the 4 in the back dunes. I recorded substrate type, presence of cover, and obstacles in 5-centimeter increments along each transect. After collecting the data, I computed the cumulative amount and relative percentage of each substrate type and if there was cover present for the transects. I did a chi-square test comparing the available habitat and the used habitat. The lab component of this study used a wooden racetrack that I filled with sand, a mix of sand and debris. I collected 22 race runners from Sullivan's Island and recorded them running on the track and quantified peak velocity and acceleration and the amount of time they ran bipedally or on 2 legs. I statistically tested the significance of sex, snout vent, and tail lengths on velocity and acceleration, and then used mixed effects and linear models to determine how impactful substrate type and gait were on peak velocity, peak acceleration, and percent of time the lizard ran bipedal. And for the field data component, I used previously collected data of lizards in the field and determined their running speed and how often they ran on each substrate. So to answer my first question, do race runners move through their habitat randomly or non-randomly? I looked at the habitat assessment and field data. Race runner habitat contains the most debris, followed by mixed and sandy substrates. Cover was mostly associated with debris substrates. And the field data showed race runners ran on sand the most in the wild and on debris the least. So to answer my second question, how available habitat affects race runner locomotion, I looked at the lab and the field data. So substrate and gait significantly affected race runner velocity. They ran fastest on debris using both quadrupedal and bipedal gaits and ran fastest on all substrates using a bipedal gait. Neither substrate type nor gait affected acceleration. Even though the difference in percent of time the lizards ran bipedal was not significant between substrates, there was a trend of race runners running bipedal on debris almost twice as often as they did on mixed and sand. And the field data showed race runners ran slowest on debris and that their speed was significantly affected by crossing obstacles. And morphological and sex data did not significantly affect either lab or field data. So the majority of race runners' movements in the field occurred on open sand, even though the most common substrate in their habitat was debris. This suggests race runners non-randomly choose to run on certain substrates. Substrate type significantly affected peak velocity during the racetrack trials, with lizards running slowest on sand and fastest on debris. There was no significant difference in velocity of bipedal strides on the three substrates. However, there was a significant difference in velocity of quadrupedal strides between debris and sand and between debris and mixed. So it would greatly benefit race runners to run bipedally across all substrate types, yet we saw that they only ran bipedal on sand and mixed about 20% of the time and debris about 40% of the time. So in the lab, race runner speed increased with substrate complexity, but the same effect was not observed in the field, where running speed was actually slowest on the most complex substrate, which was debris. So six-line race runners run non-randomly on substrates in the field by choosing to run on sand more often than debris, even though they run fastest on debris and slowest on sand in the lab. Their performance in the lab does not match their performance in the field. This could be explained by habitat and escape decisions and/or by biomechanics or morphology. Future research should focus on locomotor performance on unnatural substrates, choice experiments between two or more substrates, and running lizards on multiple substrates in the same racetrack trial. And here are the citations I used in this presentation. And if you have any questions, please feel free to email me. Thank you for listening.